Hello and welcome to today's video podcast for evolution and diversity. This is our third video on bacteria and archaea. And I wanted to spend just a little bit of time talking about where we've been and where we're going to go over the next two videos. First, we began talking about what are bacteria and archaea. What are some of the adaptations that bacteria and archaea had? Then we moved on to discussing in our last video the importance of studying bacteria and archaea. Why is it important to study these two diverse domains of life? And how does it impact us? Today we're going to focus on the vast diversity and variation that we see in bacteria and archaea. And then in the next video podcast, we're going to focus on the key lineages of bacteria and archaea. And we're going to spend a little time talking about how we study bacteria and archaea in that last video. But let's go ahead and get started on diversity and variation that we see in bacteria and archaea. All right, so for the remainder of today's podcast, we're going to focus on these four objectives. First, we're going to talk about the genetic diversity and variation that we see in bacteria and archaea. Then we're going to talk about the morphological diversity and variation that we see in bacteria and archaea. And a lot of this will be a bit of a review. And then we're going to spend quite a bit of time talking about the metabolic diversity and variation that we see in bacteria and archaea. And lastly, we're going to talk about the ecological diversity and variation we see in bacteria and archaea. So let's go ahead and start talking about genetic diversity and variation. All right, before we get started, I want to remind you of the term binary fission. I've talked about this in the past, but I want to remind you about some of its features and why it presents a particular problem for bacteria and archaea. So binary fission is simply how a bacterial cell goes from one cell with its genetic material in it to two cells that have the same genetic material in it. And it does this through a process of asexual reproduction, where the cell will first copy its DNA and pull it to either side of the cell. And then it will split the cell by pinching in the middle here. And eventually it will pinch them apart so you have two different copies of the cell. So this is a very efficient and quick process for bacteria to divide in this way. The advantage is they don't need to find a partner in order to have sexual reproduction. This allows the bacteria to continue to divide even in the absence of a sexual partner. A disadvantage is that this process doesn't create a lot of new genetic diversity. In sexual reproduction, the process of making egg and sperm generates genetic diversity, and the process of mating with another member of the species generates quite a bit of genetic diversity. Yet we know that bacteria and archaea are the oldest forms of life on Earth. So there had to have been a way to generate this vast genetic diversity that we see in these two domains of life, and how that ultimately gave rise to eukaryotes. One way it does this is through a process called lateral gene transfer. And lateral gene transfer is the movement of genetic material between unicellular organisms. Now, for our purposes, we're going to be focusing on bacteria and archaea, but this can happen in other unicellular organisms, and we also see it in multicellular organisms as well, but we're going to focus on unicellular organisms, primarily bacteria and archaea today. And so this includes various mechanisms, but this is not the movement of genetic material between parents and offspring. The movement of genetic material from parent to offspring would be called vertical gene transfer. I should mention, just in case it comes up in another class, lateral gene transfer is sometimes referred to as horizontal gene transfer. Bacteria accomplish lateral gene transfer by one of three different mechanisms that I want to talk with you about briefly. They are transformation, transduction, and the third one is conjugation. All right, so I want to just briefly describe these three forms of lateral gene transfer in bacteria. But you should know that when you take genetics, you're going to learn about each of these three in much greater detail. Let's begin with transformation. So this is when there is a bacterial cell with its own genetic material in it and all the other features that we've talked about with bacteria. And in the environment, there is random pieces of genetic material. It's not going to be a whole genome because in the environment, any released DNA is going to be fragmented to some extent. But there's all this DNA that is floating around in their environment. Now, where did this DNA come from? Well, it may have come from other bacterial cells. It could have even have come from a eukaryotic cell. And of course, it could have also have come from an archaeal cell. It really doesn't matter because once the DNA is released by these cells here because they've died and their cells have lysed, meaning they have split apart, and the DNA is released, all the DNA looks pretty much the same. And so this bacterial cell doesn't care. It can take up this DNA from the environment. And remember, this DNA used to be in these different kinds of cell types, and the bacteria will take it up. And once it's taken up into this bacterial cell, the DNA can recombine into this bacterial DNA, its chromosome. And to briefly show you what I mean here is you would have now a bacterial cell that I've drawn larger just so I could show the DNA in it, not because it actually got bigger, but it will now contain this DNA that was taken up from the environment. Now, something that sets this apart between transduction and conjugation is that with transformation, it can pick up any DNA from any species. Now, the ability to pick up DNA from any species really adds to the evolutionary tool chest for bacteria, because they're not limited to only picking up DNA from the same kind of species. The ability to do this really increases the amount of possible options as far as possible DNA options bacteria have to speed along evolution. Okay, next I want to talk about transduction. We're also going to start with a bacterial cell here with its DNA here. And transduction requires a type of virus, and we're going to call this virus a bacteriophage. This bacteriophage attaches to the outside of the bacterial cells and inserts its DNA into the bacterial cell. This DNA from the bacteriophage includes all the instructions, the genetic instructions to build new bacteria. Then that bacteriophage DNA will become inserted into the genome of the bacterial cell. Now what happens is this DNA can begin to build new viral particles, new bacteriophage. And it will make a lot of these in the cytoplasm. I'll just put times many, right? So lots of these viral particles will be made. When they fill these up with the genetic material again, they will often contain some of the bacterial DNA. So I'll just show it that way. Now what will happen is that this bacterial cell will lyse, it'll die, because the bacteriophage forced this to happen because it's so full of these new bacteriophages. And it will release these bacteriophages that has the DNA necessary to make new bacteriophage, but also some DNA from this bacterial cell. So now what happens? Well, now what happens is that this bacteriophage that has its own DNA necessary to make new bacteriophage plus DNA from this bacterial cell that has died, it will now infect a new bacterial cell. I'm drawing in a different color here just so I can show the difference in the DNA that's being moved over. But remember, it has to be the same species. I didn't say that before, but I should say that. So if this is an E. coli cell, this has to be an E. coli cell. If this is a Staphylococcus specific cell, then this has to be the same species as Staphylococcus. So now this DNA is going to be inserted into this new cell. Same species, but a new cell. And so you'll have the bacteriophage DNA inserted in here, but also some of the DNA that was in this bacterial cell that is now dead. It will now incorporate into this new cell. And now the chromosome will look something like this, where it has its own DNA that it originally had, but it's also going to have the DNA from, or at least some of the DNA from this cell here, the cell that has died. And in doing so, this bacteriophage is allowing new genetic material to be transferred from one cell to another cell. And sometimes that DNA that's being transferred to this new cell provides some sort of adaptation that this cell didn't initially have. So in this form of lateral gene transfer, DNA is moved by a virus from one cell to a new cell, and it has to be the same species. Let's now talk about our third form of lateral gene transfer, and that is conjugation. So let's start off with our same bacterial cell with its DNA in it. Conjugation allows for the transfer of DNA from one cell to another cell, which I'll draw over here. I'm drawing these in different colors, but just like we saw with transduction, they have to be the same species. And what happens in conjugation, a structure forms between these two cells, which we've briefly talked about in the past. One of these cells will have the ability to form a sex pilus, and we'll just say it's the blue one. It can form this sex pilus because this bacteria has a very special plasmid in it that has a gene that codes for the production of this sex pilus, and this cell does not. And without going into great detail, as this chromosome here is copied in this cell here on the left, it will allow for the transfer of some of that DNA over here. And in doing so, this cell now can receive some of the DNA from this cell here on the left into its own genome. It will also contain the plasmid that allows for it now to form a sex pilus. Now there are many steps between this initial conjugation event where DNA is inserted into this cell that can lead to this cell here. And you'll learn about those events when you take genetics. But for now, what I really want you to know is that this is a way that bacteria will transfer DNA from one cell to another to allow that cell to eventually to have new DNA in its chromosome. And as is true with the other two types of lateral gene transfer, this introduces new genetic material that could provide new adaptations for the receiving bacterial cell. All right, now let's move on to morphological diversity. Now within all the different species of bacteria and archaea, we see many different morphological differences. And I just want to review some of these, and many of these we've already talked about. So let me first list them, and then I'll say a few things about each of them. The first is size. The second is shape. And the third is motility. And the fourth is cell wall. In thinking about size, it's easy to trick ourselves and just assume bacteria are all the same size, and that size is very small. But there is an incredible array of sizes within bacteria. And to talk about two extremes, let's talk about the smallest first, and that is mycoplasma. And it has a volume of 0.15 cubic micrometers. Extremely, extremely small. And then the largest is a species known as Thiomargarita nambiensis. And it has a volume of 200 times 10 to the 6 cubic micrometers. You could fit, I don't recommend doing it, but you could fit over 1 billion mycoplasma into this very large bacterial species here. Now for quizzes and exams, you don't have to worry about remembering these two names, but you should be able to tell me that there is a large size distribution ranging from 0.15 cubic micrometers to 200 times 10 to the 6 cubic micrometers. We've already talked about shape, but just to remind you, there is a wide variety of shapes within bacteria and archaea. We mentioned that bacteria and archaea could be circular, so you could think of these as spheres, and scientifically we often call these cocci. Bacteria also can have the shape of a rod, and we often refer to these scientifically as bacilli. And lastly, we said they could be curved rods, and we often refer to these as being spirals. And within these shapes here, they can be arranged in clusters or chains. So now let's talk about motility. And motility can lead to different morphological differences between different species of bacteria and archaea. For instance, they could have a flagellum. But not all bacterial and archaea cells have these flagellums, so there are some differences. Sometimes bacteria can move by gliding. We don't exactly understand the molecular mechanisms that allow for bacteria to glide, but we know they can glide. And lastly, cell wall. And we spent quite a bit of time talking about cell wall, but just to remind you, in bacteria, cell walls can lead to differences in morphology based upon how much peptidoglycan is present, as well as some other molecule. But this largely distinguishes bacteria from being either gram-positive or gram-negative. Archaea, remember, they're not gram-positive or gram-negative because they don't have peptidoglycan in them, but rather they have this S layer, which is just a layer of proteins. All right, this is all I want to say about the morphological diversity that we see within bacteria and archaea. All right, we've talked about ways that bacteria and archaea have and generate genetic diversity and variation, as well as some morphological diversity and variation we see in bacteria and archaea. Now I want to talk about metabolic diversity and variation that we see in bacteria and archaea. Now it is true that all life have very specific metabolic needs, and because of that, there's quite a bit of diversity within all life forms. However, the metabolic diversity and variation that we see in bacteria is far greater than we see in any other life form. Remember that all life has two nutritional requirements. The first is that they need chemical energy in order to make cellular energy. And usually that cellular energy is in the form of ATP. And the second is they need a carbon source to make macromolecules. Like DNA and RNA, proteins, lipids, and carbohydrates. Let's first begin with the three different ways that archaea and bacteria acquire chemical energy in order to make cellular energy. All right, so let's address this first nutritional need by all organisms. And in bacteria and archaea, they acquire this energy to make cellular energy in one of three different ways. And we're going to describe these three different ways by classifying bacteria and archaea in one of three different groups. And these three groups are, first, phototrophs. The second group is chemoorganotrophs. And the last one includes the chemolithotrophs. Phototrophs use light energy to excite electrons. This results in the production of ATP. And so what we're talking about here is largely photosynthesis. So let's talk about chemoorganotrophs next. And I'm going to need a little bit more room, so I'm going to erase chemolithotrophs for now. Chemoorganotrophs, they oxidize organic molecules with high potential energy. These molecules are often sugars, like glucose. From this process, ATP is produced by cellular respiration. Alternatively, we could also produce ATP in this way by fermentation. You should recognize that chemoorganotrophs are found in all three domains. We see these in bacteria, archaea, and eukaryotes. All right, let's talk about the third one next. And the third one, remember, were the chemolithotrophs. And chemolithotrophs, they oxidize inorganic molecules with high potential energy. Some of these inorganic molecules with high potential energy include ammonia or hydrogen sulfide. Now, this is very similar to what we saw with the chemoorganotrophs in that we're still using cellular respiration. But inorganic molecules serve as electron donors. The chemoorganotrophs, remember, they use carbon-based molecules as electron donors, such as sugar. Okay, let's move on to this second nutritional need for all cells, and that is the need to obtain carbons to build larger macromolecules. Bacteria and archaea achieve this second nutritional need by being either autotrophs or by being heterotrophs. Autotrophs synthesize their own compounds from simple starting materials such as carbon dioxide or methane. So these are self-feeders. This would include organisms like plants, but it also includes organisms like cyanobacteria and methylomonas. So if autotrophs are self-feeders, then heterotrophs are other feeders. They absorb ready-to-use organic compounds. These organic compounds that they absorb are things like building blocks, like amino acids, simple sugars like glucose, and nucleic acids, and other larger organic compounds. These larger organic compounds, remember, they can't make themselves, so they have to get that from other organisms. They could be other heterotrophs, or they could be autotrophs that they get them from. For instance, humans are heterotrophs. We can obtain our organic compounds by eating other autotrophs, like a cow, a pig, a chicken, or we can gain our organic compounds by eating autotrophs, by eating plants. So as I said at the beginning of this, bacteria are extremely diverse when it comes to their metabolism. And that's because they can be autotrophs or heterotrophs. Some species of bacteria can be phototrophs, chemoorganotrophs, or chemolithotrophs. Now, every bacterial or archaeal species is either an autotroph or a heterotroph, and a phototroph, a chemoorganotroph, or a chemolithotroph. So when we combine those, we see we get 6 different combinations. So as an example, photoautotrophs are bacteria that can make their own building blocks and ultimately their own macromolecules. They also gain energy from the sun. And let's look at one other example. Bacteria that are chemoorganoheterotrophs obtain their building block carbon molecules by absorbing it from other organisms. And they get their energy from other molecules, from other organic molecules like sugars. Now, I should mention that this figure is in your textbook. It is figure 26.3, and it's on page 546 in the 7th edition. So what do I want you to know from this table? Well, you certainly need to know the top 2 up here, autotrophs and heterotrophs, and you need to know these 3 here, phototrophs, chemoorganotrophs, and chemolithotrophs, just as we described on the previous couple whiteboards. You should also know that you can combine these as shown here. You do not need to know examples that are given here for each of these. But if you memorize these 2 and then these 3, you should be able to combine them in different ways to explain, for instance, what a chemolithoheterotroph is or what a chemolithoautotroph is. So focus on memorizing autotrophs and heterotrophs and phototrophs, chemoorganotrophs, and chemolithotrophs, and then you should be able to fit them all together to explain these 6 variations. Okay, let's go ahead and move on to our last topic for today's video podcast, and that is understanding the ecological diversity and variation of bacteria and archaea in our ecosystems. So I think it's an important question to ask. What is the role bacteria and archaea have in our ecosystems? It seems that they must have a very important role because we talked about how their abundance is great. We know that their biomass, that is the mass of all bacteria, is greater than all the animals and plants on Earth combined. We also know that their habitat diversity is vast. They live in almost every possible habitat. As we mentioned, they live in Arctic ice, deep sea vents, or near deep sea vents. We find them in our body, in our intestines. They're everywhere. It seems with this large abundance and diverse habitats that they must play a significant role in our ecosystems. So let's first begin with how they fit in a typical food chain. So here's a very simple food chain, and when you take ecology, you're going to go into much greater detail about food chains and food webs. But for our purposes in this class, this will suffice. A typical food chain will begin with the sun, and the sun provides the energy for autotrophs to generate their own cellular energy, but also those large macromolecules. And so let's first begin by talking about what a producer is. Essentially, a producer is an autotroph. It's an organism that can make their own food. And we know that producers, or these autotrophs, can be one of a variety of types of animals. They can be bacteria, or some bacteria. For instance, cyanobacteria are producers. They can make their own energy, and they can make their own macromolecules. We know that plants are producers. As well, green algae are producers, and some protozoa. All of these producers are critical in providing nutrients to heterotrophs. So for instance, in this food web here, a grasshopper is found, and they will consume various producers. And they are a primary consumer. Then a secondary consumer, like a frog, is going to eat the grasshopper. A tertiary consumer is going to eat the frog, such as a snake would eat a frog. And then there's this final consumer. Sometimes we call these apex predators. They will eat the tertiary consumers. Ultimately, though, all of these organisms along this food chain will die. And when they die, decomposers come along and release some of those nutrients found in these different organisms back into the soil. And some of these decomposers include bacteria. There are also various fungi and other microorganisms, but bacteria and archaea certainly play a role as these decomposers in our soils. So I want to include decomposers down here, in addition to the producers, because both producers and decomposers include various bacterial and archaeal species. Producers get the food web or food chain started, and decomposers finish it off. So bacteria and archaea have a critical role in our ecosystems from this perspective. We're going to come back to bacteria and cyanobacteria in the next video podcast when we talk about the oxygen revolution. So we're not quite done with cyanobacteria. In fact, we're going to talk about cyanobacteria on the next whiteboard as well. Okay, next I'm going to talk about nitrogen-fixing bacteria. I think it's important to remember that all life requires nitrogen. We need nitrogen to make proteins and nucleic acids. So it's very critical that all life has access to nitrogen. This seems relatively simple at first consideration because our atmosphere is about 78% nitrogen. It seems like that might solve our problem, but it doesn't. And the reason it doesn't is because the nitrogen in our atmosphere is held together by a triple bond. So nitrogen gas is into, and it's a very tight interaction between these two atoms of nitrogen with 3 bonds holding them together. Most organisms can't break down nitrogen into a usable form. So then the question is, where do archaea, bacteria, and eukaryotes get their nitrogen? Well, it turns out that all eukaryotes and some bacteria and some archaea get their nitrogen from ammonia or nitrate. So I'll write here, most get their nitrogen from ammonia or nitrate. Again, when I say most, I mean all eukaryotes and many bacteria and many archaea. However, some bacteria and some archaea can convert nitrogen gas. Remember, that's that nitrogen held together by 3 bonds. They can convert this into ammonia, NH3. And they do so because they have an enzyme called nitrogenase. So they have a gene that codes for this enzyme, nitrogenase, which converts nitrogen gas into ammonia. And then these bacteria and archaea can use this ammonia. Now, the bacteria that can do this, some of them are free living, meaning they don't form any community with other species, but some live in association with plants. And when they live in this association with plants, they can generate a lot of this ammonia that then those plants can use. I'm going to keep this down here so we can refer to it, so I'm going to erase this middle part here. Now, I didn't say it down here, but when we talk about these bacteria that have this nitrogenase that can convert nitrogen gas to ammonia, we call these the nitrogen-fixing bacteria. And remember, when they're living by themselves, not in association with any other organism, the ammonia that they produce, they can use. However, when they live in association with a plant, they can provide the nitrogen, the ammonia that is, to those plants. And so I want to talk about two quick examples of how these nitrogen-fixing bacteria live in a symbiotic relationship with plants. And the first includes our good friends, the cyanobacteria. So some species of cyanobacteria are nitrogen-fixing bacteria, and some of them can provide a nitrogen source to plants like rice plants. In providing this nitrogen to the rice plants, the nitrogen serves as an excellent source of a fertilizer. We also see nitrogen-fixing bacteria on roots of plants. However, not all plants. We only see them on a relatively few number of plants, but these include plants like the legumes. Legumes, remember, include many varieties of beans, peanuts, lentils, black-eyed peas, and various other examples. Again, when they live on these roots of these legumes, they provide an excellent source of nitrogen in the form of ammonia so that these plants can grow optimally. Now I'd like to talk about nitrate pollution. It's important to remember that not all bacteria can fix nitrogen. It's also important to remember that not all plants have nitrogen-fixing bacteria associated with them. Remember, those plants that do have an excellent source of this nitrogen fertilizer, and it really helps their ability to grow. Some plants that do not have these nitrogen-fixing bacteria associated with them include crops like corn and wheat. These are very large agricultural crops that we grow in this country and are grown in other countries. To help these crops grow optimally, farmers will use fertilizers that have a high concentration of nitrogen compounds. This is really helpful in growing these plants. However, there are some downstream consequences that are causing significant ecological problems, and I want to explain one of those to you. The first step that happens is that a fertilizer with ammonia in it, high concentrations of ammonia, is used on crops. And let's say this is happening in North American farms in the Midwest. And let's talk about corn in this example. And so corn uses some of this ammonia to increase overall yield. However, most of that NH3 is not used. The leftover ammonia is used in anaerobic respiration by some bacteria and archaea in the soil. This in itself may not seem like a necessarily bad thing, but it leads to a negative consequence. And that is because these soil microbes, when they use NH3, they produce a byproduct of nitrate. And so why is nitrate a problem? Well, let's talk about that next. Nitrate enters the groundwater under the soil. This will then run off eventually into, let's say, the Mississippi River. And then that will run off into the Gulf of Mexico. This then leads to an increase in growth of marine algal and cyanobacteria. This increase in the marine algae and the cyanobacteria we call a bloom. Eventually, these cells that bloomed here, they will die. So the cells from number 4 here will die. Sad but true. Then decomposers grow rapidly. And that's what they're supposed to do. They're supposed to decompose dead organisms, like these marine algae and cyanobacteria. However, these use lots of oxygen. And in doing so, they create large dead zones in the Gulf of Mexico. Right now in the Gulf of Mexico, there is a dead zone, meaning no life is living there at all. And this dead zone is about the size of the state of Connecticut, because these decomposers are using up all of that oxygen. And it just gets worse and worse and worse over time as more nitrogen is used on the farms and more nitrate is released into the groundwater and into the Mississippi River and ultimately into the Gulf of Mexico. Because of their diversity, because of their abundance, because of the many different habitats they live in, bacteria and archaea have huge ecological impacts on our ecosystems. That is all I have for this video podcast. If you have any questions at all, please make sure you reach out to me. If not, I will see you in class. Bye for now.